Sorry, got ahead of myself, but thank you very much. Was that you, Abigail? No, Alyssa. Thank you for that. I appreciate it. If I forget to record, please do so because sometimes I will. Unfortunately, WebEx doesn't do it automatically. All right. Ah, crap. That's annoying. Well, at least we only missed like 24 minutes of it. I'll put out an announcement. Oh yeah, sorry. Also begin with A. I'll put out an announcement about the exam and next Tuesday, just so everybody has it kind of on the record, so to speak. So anyway, alleles, right? Different versions of those factors or genes. Second one is that those alleles segregate independently or randomly. Randomly is probably a better word. There's a whole bunch of randomness to sexual reproduction, which is really the point of sexual reproduction, right? And so if you have two different alleles, right, and you can only have maximum two versions in one individual because we're diploid, right? We have two copies of each chromosome. So which one goes into which gamete, sperm, egg, spore, if you're a fungus type thing, which one goes into which gamete is random. So if you have two different version ones, it could be, let's do like short finger. Long finger goes into this one, short finger goes into this one. Or it could be the other way around, right? That's random. That's really what the monohybrid cross is all about, right? In terms of illustrating that principle. The third law is that if you have two genes, right? This gene does not affect which allele goes into the gamete of this gene, right? So they act independently, right? So if you have, oh, this is going to get awkward, right? You know, this allele could go into the gene and this one, that one, or it could be this one and this one, right? Doesn't matter. Those two genes behave independently. So essentially, the law of segregation, which says, you know, if you've got two different alleles, which gamete gets which allele is random, the law of independent assortment is basically taking that and saying that's the same for all genes. Right? We know that's not actually the case, right? There are exceptions to that. There's a lot of exceptions to that. But that was the kind of founding law, right, that we work with. And a lot of what Mendelian genetics is about is essentially using these different crosses, right? And there's three that we're going to talk about: monohybrid, dihybrid, test cross, right? And the test cross is actually pretty important for a lot of different reasons, right? But it's basically we have a whole range of assumptions that we're going to be using those to test, because a lot of those assumptions are unrealistic, right? They don't really affect what's going on in real life. But if you want to figure out what's going on in real life, you have to start from a series of kind of real basic assumptions, right? And the assumptions are sex is random, or fertilization, reproduction, all of it's random, right? Super random, randomness all over the place, right? Or randomnosity. Random, sex, reproduction, fertilization, whatever, right? So one sperm doesn't get the edge over the other, even though sometimes they do. But that's our assumption. Sex is random, no mutation, because we don't want new alleles kind of creeping in the middle of a cross, because that would just totally screw everything up. Oh yeah, no selection, right? So there's no external factor which will affect the outcome of that cross, whatever it might be, right? So random, no mutation or no new alleles, and no selection, right? So those are three kind of generic ones, right? The fourth one, not very good at doing this, is that those alleles segregate randomly, right? For any particular gene, whatever alleles you have, they're going to end up in different gametes on a random basis, right? So if you have two different alleles, you get a 50-50 chance of getting one or the other, right, in a gamete. And then the fifth one is that what alleles you inherit for one gene has nothing to do with what alleles you inherit for the other, right? Those are the 5 assumptions. Random, no mutation, no selection, random segregation of alleles, which kind of comes back to this one, and one gene does not affect another, which again comes back to this one, right? So those are the 5 things that we're going to be working with. And so the monohybrid, dihybrid, and test crosses always have this very kind of rigid structure. And if they don't follow that structure, they're not one of those crosses, right? So whatever kind of cross, you could use whatever trait, whatever alleles, whatever, right? Doesn't matter. If I say it's a monohybrid cross, you know it's always going to follow a particular structure, right? Same for dihybrid cross. Test crosses are different. So let's go back over here. So this is always, I don't know why it's P1 in US. I'll have to look into that. I was always taught it was P0. Doesn't really matter. P, whatever number after it. You're always going to have this for monohybrid or dihybrid crosses, these 3 generations, and they always have a particular specific makeup, right? The first generation, the parental generation, is always between 2 true breeding parents. And what true breeding means, there you go, up at the top, right? Basically means that if you take that one parent and you breed it with another one exactly like it, you'll only ever get the same phenotype out, right? So if you cross a red plant with a red plant and they're true breeding, you'll only ever get red plants or red flowers or whatever, right? You'll never get a white flower or purple flower or, any other color. It'll always be exactly that. And what that really means, even though this often applies to horses and European royalty, is that they're homozygous, right? So that means that the parents are always going to be homozygous for any given gene. Now, typically, they're going to be the opposite, right? So we're going to have one which is homozygous dominant, one which is homozygous recessive. When crossed together, they will always produce heterozygous offspring called the F1 generation. Always. There's nothing else that they can do. As far as I can aware, I've never thought of, no, I don't think they can, right? Because that's it. And this is called the F1 generation, right? So these are always heterozygous, and really importantly, I can't stress this enough, the phenotype of the F1 generation will be the same as that of the homozygous dominant parent, because they will both have one, at least, dominant allele, right? And so they'll express the dominant phenotype. Okay, so that's actually a handy trick you can use to figure out which of those parents is expressing the dominant phenotype. All you have to do is cross them together and look at the phenotype in their offspring. Whatever the phenotype in the offspring you see, that's the dominant phenotype, right? I mean, it's kind of makes sense when you think about it. And then you will cross those F1 individuals with each other, called selfing, either directly with themselves, or if they're not plants, then with their siblings, basically. And you'll get the F2 generation. And this is where you see that ratio, either a 3-to-1 ratio for monohybrid crosses or a 9-to-3-to-3-to-1 ratio for dihybrid crosses, right? And when we talk about those ratios, we talk about phenotypes, right? The phenotypic ratios. Okay, anybody got any questions? That's a good place to take a pause. Found some more coffee. There's actually a funny XKCD comic where the joke of the panel is basically 2 geneticists go on a date, and the first thing they do is, when they sit down for dinner, is work out a Punnett square of what their offspring will look like. Yes, I always found that funny. I should go and dig it up, find it somewhere and post it. Okay, actually, while you're thinking about anything about that, let's bring this up. I'm just bringing up, oh, come on, you pain in the ass. Blackboard, so I don't forget to post those announcements after class. "101 clicks later." All right, so I'm just doing this as a place marker because I have a terrible memory. And spelling too, by the looks of it. All right, so I'm just going to leave that there. Let's get back to this. All right, so any questions? If not, don't worry. But, if they occur to you, stick your hand up or shout them out because I want to make sure you get this stuff. Okay, so let's scooch back up to here. All right, so my hope is that this is just really a recap for all of you, right? So this is really going over stuff that you have already covered, probably in high school, or if that was many moons ago for you in Gen Bio 1, or if even that was a long time ago for you. I don't know, well, you get it now, right? Yeah, high school, huh? What a distant memory, if a memory at all, right? When you get old enough, you just start forgetting stuff. Anyway, so this is a monohybrid cross of the classic round versus wrinkled peas. Actually, when I do this in person, I prefer to use cats because they're a lot more fun to draw, and cats are just cute, right? But anyway, that doesn't matter. Kind of wistfulness. I'm actually really good at drawing cats. Let's see if I can draw a cat. A little bit of a random. It's all right. I'm just getting the lid off my pen. I've got a very jumpy dog. There you go. It's a stylized cat. Anyway, that's a white cat. Drawing white cats on whiteboards is really tricky because obviously you just say, look, there's a white cat. Anyway, a little bit of a tangent there. What's up? You can come up? No. She's having a stretch instead. So anyway, so this is a good example as any of a monohybrid cross. You got your P1 or P0, depending on what you want to call it, generation, your F1 and your F2. So really what we are interested in is the F2 generation, right? That's where we see the effects of that random segregation of alleles and random inheritance of those alleles, right? So that's a really good example of both of those things, right? And when we get to meiosis, we'll actually see the underlying cell biology or even molecular genetics of that randomness, right? It's actually pretty neat to think about. So in our P0 generation, we've got both homozygous, right? So we've got homozygous round seeds, homozygous wrinkled seeds. Now again, remember what I said, you don't actually need to know which one of those which one of those phenotypes is dominant, right? Because all you do is you cross them together and each one of these plants, not the seeds themselves, obviously this is just a phenotype, each one of those plants can only contribute one type of gamete for that particular trait because they're homozygous, right? They only have one type of allele in their cells. So whether they're dominant or recessive, each gamete gets the same allele, right, of that individual. Now, when you cross those two together, essentially you're going to have a gamete with a dominant allele and a gamete with a recessive allele, and then those come together to form the diploid offspring, the F1 generation, right? So if you have a dominant and a recessive, now you have a heterozygous offspring, F1 generation, and you have an offspring where they have at least one, well, they have one dominant allele. So whatever phenotype you see in the F1 generation is the dominant phenotype because they're heterozygous. And that applies to a dihybrid cross too, which we'll get to in a second. So if these are round, that tells you, going back up here to the parental generation, that the individuals with round seeds are homozygous dominant. And so one thing that, I don't know whether I'll do it this exam or not, is I'm quite fond of basically giving you a partially filled out monohybrid cross. And I expect you to know the genotypes or work either up or down, not sideways, and fill in those blanks because it's always exactly the same. There is no variation in a monohybrid cross. If there is, it's not a monohybrid cross anymore, right? So this is one way in which you can use that kind of knowledge of what's going on here and apply it to work out what's going on up here or potentially what's going on down here, right? So that's worth remembering. Now, down here, we're going to have essentially the offspring of what happens when you cross two of those individuals together. And I can't just do this with words because this would just be painful. Right. Unfortunately, kitty cat is going to get scrubbed. Perhaps it's still there, right? Because it's a white cat. So if we have an individual that is heterozygous, let's use HH. I like using letters which have very different upper and lower cases. Right, and so the F1 generation are going to be selfed, so they have the same genotype. Right, what we really care about is what gametes can these individuals produce, right? And it's either going to be gamete with a dominant allele. Can you enlarge U on the pic? Oh, okay. Hang on. Gar. Get away from there. Sorry, I have to wait for a panel to disappear. Let's stop sharing this. And that should blow me up bigger. Does that embigorize me? Awesome, cool. Okay, thanks for a reminder. If I forget in the future, which is a good chance I will do, just remind me again. And the other possible gamete is going to be a recessive allele, right? And you're going to have the same for this one as well, because it has the same genotype, right? And so the gametes are what we care about because it's the gametes that will recombine to produce the next generation, right? The offspring of the next generation. So that's what we have to focus on. The genotype of the individual in the cross, like this one or this one, that knowledge really is there to tell us what gametes they can produce. Because again, it's the gametes that matter, right? It's not, who you are, it's what gametes you can produce, and we'll combine with someone else's gametes to produce another individual. And so that automatically gives us our Punnett square, right? And the reason why I'm going through this in detail, you may think this is like, yeah, sure, I can do a monohybrid cross, and that's good, right? It's because you apply these exact same principles to any cross. Dihybrid, test cross, doesn't matter. And you could even do a trihybrid or a quad hybrid or a pentahybrid cross if you want, if you got, too much time on your hands. And you can just work it out, right? It's the same principles. The same things apply. It's just a lot easier to teach them using a monohybrid cross because it's simpler. There's only one gene and two alleles involved, right? And so really what you're doing with a Punnett square is you're doing like an on-paper version of reproduction, right? This individual is going to produce a whole bunch of gametes, blah, whatever they may be, spores, let's say, if you're a little squeamish about biology. And this individual is going to do the same, right? And then actually, if you're a giant clam off the Great Barrier Reef, right? You know, they're pretty cool. They are freaking enormous, like huge, big as my dog. And she's a pretty big dog. They literally do that, right? So they like puff up, you know, a stream of basically semen. It's kind of best not to think about that when you're swimming around in the sea, what other things are in there, because it's kind of gross if you do. But anyway, they literally eject a stream of semen, right, gametes, and those kind of float around in the ocean. And later on, you know, it will say, okay, don't want to do that no more. And they'll change to producing a stream of eggs, essentially, right, the other sex gamete. And so those literally do just kind of float around in the water column, you know, get stuck to tourists and, you know, get eaten by other things and stuff. But they will then recombine randomly with the matching gamete, ideally from another giant clam, to produce little clamlets, right? I don't know what baby clams are called. But it's totally random, right? So they're really putting out all of these gametes, and those gametes have every single combination possible, most likely if there are enough of them, of all the different alleles for all the different genes that that clam has. And so when you do a Punnett square, really you're just kind of simulating that process, right? Assuming that there are equal numbers of each gamete around, right? And so the point then is that you just add them together, right? You add the gamete from this individual to the gamete from this individual. Sorry about the kind of mic noises from my fan over here. And then you do the same over here. Now, if you're a pedant, you'd do this one the other way around, but it doesn't matter. And that's how you get the F2 generation. So remember, these are the F1 generation. And let's use a different color because it makes life a little bit easier. All of these are your F2 generation, right? So these are what we're counting. These are what we care about when we're doing a monohybrid or dihybrid or test cross or anything like that, right? And so then I'm going to use the same color just because I don't know where my blue pen has gone. Right downstairs somewhere, I guess. Now you're going to look at phenotypes, and what you care about for phenotypes is the presence of a dominant allele. If you have a dominant allele, you have the dominant phenotype. If you don't have a dominant allele, which the only ones that don't are these homozygous recessive, then you'll express the recessive phenotype, right? And so that's what gets us our 3, 2, 1 phenotype ratio. That looks so weird, kind of backwards on my thing. Hopefully it looks right to you lot, right? That's how you get that, right? So every monohybrid cross, I'm gonna put that down because my arm's aching now. Every monohybrid cross is exactly the same, right? Even if obviously if the trait that you're looking at differs, which one hopes it will. Don't want to work with peas forever, right? Or the alleles or the phenotypes or whatever, you'll always have exactly the same outcome. 3-to-1 ratio dominant to recessive phenotype. Now that should be all pretty straightforward. But I'll give you a sec I'll just bring up the PowerPoint again to cogitate on that. So if you got any questions, thoughts, clarifications, have a think about that and let me know. And again, if I forget to maximize the image of me, just let me know and I'll do so. Anybody got any questions? Questions? Okay, we're not going to go into reciprocal crosses because we talked about that already. Okay, so one thing that needs talking about is the test cross. I better get this off. Did anybody notice I actually cleaned my whiteboard? Look at that, isn't that beautiful? Super shiny. Okay, let's get rid of this again. So before I get onto that, essentially the deal behind the test cross is it comes back to the issue of that dominant allele. So if you have one dominant allele, you express the dominant phenotype. So if you have an individual expressing the dominant phenotype, you cannot figure out its genotype just by looking at it, because it could be the homozygous dominant or it could be heterozygous, both of which produce the same phenotype. The only phenotype for which you can tell what the genotype is the recessive one, because that has two recessive alleles. It's homozygous recessive, right? So if you want to figure out what the genotype of that dominant individual is, the only way you can do that is by doing a test cross. So let's say you have a dog with blue eyes, right, which is the dominant, I don't know whether it is or isn't, let's just use this example. You have a dog with blue eyes, and blue eyes is a dominant phenotype. You want to breed that dog, right? But you want to make sure that whatever offspring you produce are going to have blue eyes, because those are, I don't know, let's say super valuable, right? And so you want to really make sure that you've got the dog that you want in terms of breeding. So the question is, how do you figure out whether or not that dog is homozygous dominant or heterozygous? And the way you do that is by using a test cross. And so a test cross takes that kind of unknown individual and crosses it with a known homozygous recessive individual, dog in this case. Right, so let's stop that. Yes, I do want to do that. All right, so I should be invigorized, hopefully. Okay, so essentially you're going to have two possible crosses here, because you have two possible genotypes, right? So if you're, let's do B. Actually, let's do a homozygous dominant first. Right, so if your cute fluffy dog is, this is blue-eyed. Right, forgive my terrible handwriting. It doesn't get any better when I'm doing it on the couch. Right. If you have this, and we're going to cross it with, and again, I don't know whether blue-eyed is dominant or not. It's just an example, right? Let's see, you should be able to see that. That's what a test cross is going to look like. So in that case, you're going to get only dominant alleles from your unknown individual. And because your tester, right, the thing that you're testing against is homozygous recessive, all you're going to get from this individual is a recessive allele, right? So you Make those together. All right, question 2, class. What is your phenotype going to be? Blue eyes.
Blue eyes.
Oh, yeah, loads of people are. Sorry, I just saw all those answers come up in the chat room. Sorry. Right. And how many different phenotypes are you going to see in this generation? It's just going to be one, right? So if you have one set of offspring, right, you only ever see blue eyes from that this mating. Let's say you get like 8 pups or whatever. You're going to only ever see 8 blue-eyed pups. That's it. So if you get that, this tells you that you have a homozygous dominant individual. Now, again, we don't know if it is or isn't until we do this. So what's the other possible outcome? This is going to make... Oh, I could practice doing genetic crosses with my left hand. This is going to look bad. Right, so now we've got, this is the other possible option, right? So this is still going to be, oops, blue-eyed, right? But instead, this individual can produce 2 gametes, or 2 types of gametes. Yeah, does that make sense? So when you cross this individual, and these are going to be random, right? Again, you're going to get 50-50 big B versus little b, right? Because this process is random and it has to be, otherwise this doesn't apply anymore. And so when we cross these together, right? Half the individuals are going to be blue-eyed, heterozygotes, exactly the same as we get over here. But half of them are going to be homozygous recessive. And these will have this homozygous recessive phenotype, the homozygous recessive, the recessive phenotype there, right? And because these alleles get produced at random frequency, right? So it's going to be 50-50, big B versus little b. You're going to get a 1-to-1 ratio of blue to brown puppies. So let's say if you have 8 pups, most likely, give or take, right, because nothing's ever super precise, you'll get 4 brown and 4 blue-eyed puppies. And if you do, you get this. That tells you that your individual is heterozygous, right? And so that is what a test cross is. Anybody got any questions? Where's my black pen lid gone? Does that make sense? If it doesn't, now's a good time to... Damn it. I think I'm sitting on my pen lid. Oh well, I have to find that afterwards. Anybody got any questions about test cross?
Professor, can you pull up the board again? I wanted to see.
I'll try and move my laptop.
On the lower part, really can't see too much. So you're saying it's 1-to-1 because we crossed the dominants with the new hetero?
So the 1-to-1 is because the test this homozygous recessive individual here, this is our tester in the test cross, that only ever produces lowercase b alleles, right? But if the unknown is heterozygous, then it's going to randomly produce a 50-50 ratio of dominant gametes versus recessive gametes, or gametes with a dominant allele and gametes with a recessive allele. It's 50-50. Does that make sense?
Yes.
So because this is a 50-50 ratio, on average, right, if you look at enough offspring or gametes, when these gametes are randomly combined with the gametes from our tester, you're going to get a 50-50 ratio of homozygous recessive to heterozygous individuals. And because these two genotypes have different phenotypes, now you're going to get, is that pointing to blue? Yeah, no, that's pointing to brown. You're going to get a 1-to-1 ratio of brown-eyed to blue-eyed individuals, because you have a 1-to-1 ratio of the dominant to recessive alleles from this heterozygote. Really, in many ways, the contribution of the tester doesn't actually count, because if you get a recessive allele from the heterozygote, you're going to have the recessive phenotype. If you have the dominant allele from the heterozygote, you'll have the dominant phenotype. So this is really just kind of providing a kind of blank canvas, essentially, on which you can see which of these alleles you're getting, or even if both of those alleles are present. Because if they're both present, you'll get those two different phenotypes. If you only have that one present, you'll only ever get one phenotype. Does that help?
Yes, thank you.
Yeah, so really when we talk about the ratios, what we're talking about are the ratios in the test cross, at least, right, of the alleles that were in the unknown individual. Right, and so that's basically what you see in the offspring. It's a little different for mono and dihybrid crosses, because those ratios reflect the different combinations of alleles you can get in the F2. Right, so that's kind of one difference between the two of those. Okay, good question though. Oh, found the lid. I was clever and put it on the end of my pen. Okay, so let's have a, oh no, I don't want to do that. I want to do this. Let's go back here. So really a dihybrid cross, I want to look at the dihybrid cross now. A dihybrid cross is exactly the same as a monohybrid cross, pardon me, with one exception. So this is one trait. We're interested in this case, P shape, I guess I would call it. And that's controlled by one factor or one gene. And that gene has two different alleles, right? So that's a monohybrid cross. Dihybrid crosses are exactly the same, but we have two genes. All right, so now, and two traits essentially, right? So now we have, again, the same P kind of structure trait, round versus wrinkled, and we have seed color. Right, now I'm being really careful to say these as traits, right? Seed color is the trait, the actual color is the phenotype, right? So if we have seed color as the trait, then yellow and green are the two different phenotypes for that trait. It's kind of a tricky distinction, but it's an important one to stress. Now, the same principles apply. The P generation are going to be true breeding homozygotes. So they're going to be homozygous dominant, homozygous recessive. Now we typically write this as one side of the, well, one parent being homozygous dominant for both, and the other one being homozygous recessive for both. It doesn't actually matter. You could have one and one, like one homozygous dominant, one gene, one homozygous recessive, and the opposite on the other individual. We just write it like this because otherwise it's just a little, could get complicated. And so the same principle applies as with the monohybrid cross. I'm just going to kind of scrunch this off in the meantime. You always have to think about what gametes can these individuals produce. That's really the key, because again, it's the gametes that recombine randomly to produce the offspring of the next generation. So those gametes are what we care about. And so when you have homozygous anything, you're only ever going to get a single type of gamete, right? So I'm going to do this with a screen small for now, just because switching back and forth takes a fair bit of time. So if you have your, I'll write it real large. This is round and yellow. All that can produce, gamete-wise, is that. Nothing else. Right? Because you're only ever going to get your dominant R allele and your dominant Y allele in your gametes, because there's no other alleles there, right? Now, one thing that students do, which I don't fully understand, is they often write this as that. 2 R's. That can't happen because otherwise you've lost the chromosome that the Y gene is on, right? So always make sure you have one allele of both genes in your gametes. If you've done that and you account for all the possible combinations, you can't get it wrong. In this case, there's only one possible combination because you're homozygous for this and for this. Now, don't forget, we could actually do a test cross with this, right, to see whether or not this is truly the case. I'm not going to go there today because it might be a wee too much. So if you can only produce dominant-dominant and let's make sure that's as big as possible. Recessive, when you cross those together, all you're going to get is a heterozygote. And it's going to be heterozygous for both genes. Okay, so before we skip on to the next slide, you can probably do this yourself if you want to look ahead. What phenotype is that F1 individual going to have? Let's bring up round yellow. Yay, totally. If I could throw you a chocolate, Angela, I would do it. It'd have to be like a virtual one or something. Or a caffeine treat probably would be more useful. Okay, so that's what you're going to get in the F1. Now things get complicated. I actually used to have espresso sweets. Man, those were really effective. Kind of have to swear off them a little bit, otherwise stuff starts getting a little crazy. So this is our F1 generation, and you have to work out, so this is a little bit crazy, and I'll stop sharing so I can kind of write this out bigger. You have to account for all the possible gametes you can get from that individual, right? So let's stop sharing this quickly so we can get this a little bit bigger. Now I don't have space to write all of this out on here because it's tiny, and I'd have to write it really teeny tiny too, so I'm not going to do that. But what I'm going to do is I want to just stress how you get those 4 different combinations. And again, if this is all really straightforward for you, that's awesome. This is really just to make sure everybody that doesn't completely get it does so by the time we're finished. Okay, so this is our, oops, this is our F1, right? So it's heterozygous for both seed roundness, whatever you want to call that, and also seed color, right? So this is going to be round and yellow. Okay. Now, the gametes from this can only have one gamete from each gene, one allele for each gene. But we have 2 different alleles for each gene, right? So that basically tells you you're going to have 2 times 2 combinations. You're going to have 4 total possible gametes. Now, there's no, how can I put it? When I say possible gametes, if you only look at one gamete from this individual, it could be any one of those, but you get a 25% chance of finding one in particular, right? If you look at enough of them, and you obviously do that by looking at the offspring, you should get these in a 1-to-1 to 1-to-1 ratio, right? 25% of all the gametes produced will have this one, 25 this one, 25% this one, and so on, right? Because it's random. But you have to account for all of them, because otherwise it doesn't make any sense, right? So we're going to have big R, big Y, right? That's one of them. And you can do this whatever order you like. It doesn't actually matter. I just have a very particular way of doing it just from habit, right? You can have big R, little y. You're going to have little r, big Y. And you're going to have little r, little y. Right? So those are the 4 possible gametes you can get from that one individual. And these are going to be in a 1-to-1-to-1 ratio, right? You're not going to get more of one than the other, at least, based on probability's sake, right? So that's why when you do a Punnett square for a dihybrid cross, pardon me, you're now going to have 4 columns, 4 rows, right? So let's get back to this. Let's point this fan a little bit. Yeah. It's right, dog. Just wondering what I'm doing. Probably wondering in general, I don't think dogs do genetics very well. Anyway, so now when you look at this Punnett square, now you have 4 possible gametes along the top. And they do it in a different order. It doesn't actually matter too much. And because the F1 generation is selfed, so each individual in this cross are going to have the same genotype, right? They're going to be heterozygous for both genes. You get the same set of alleles going down the bottom. And then all you do is you just add them together, right? For example, the top left square, you take the big R from that gamete and the big R from the other gamete, you got RR, big R, and the same for the other gene. And you just go through that and do that. And then what you do, and this is where the important bit comes in, you scan through all of these and look at whether or not they have a dominant allele for each gene. If they do, they will express the dominant phenotype for that gene, right? So you can see that this individual here, right, homozygous dominant for both, expresses both dominant phenotypes. This one over here down towards the bottom right is heterozygous for both, expresses the same 2 dominant phenotypes. And all other combinations that have one at least one dominant allele for both genes. Now you'll also see down here we've got a phenotype ratio. And so essentially you have two different traits, two different genes, each with two different phenotypes. And so it's the same, 2 by 2, you now have 4 different phenotype combinations. But because of the way the law of dominance works, oh, there you go. That's the first law of Mendel. It's the law of dominance. I can't believe I forgot that. The way the law of dominance works, you don't get an equal proportion of those phenotypes. In fact, you get a really skewed ratio of 9 to 3 to 3 to 1, where the vast majority are, well, not vast, but majority of the phenotypes, the individuals, have both dominant phenotypes. And the very small minority has both recessive phenotypes. Now, this ratio, and I can't stress this enough, is really important. It's not important, well, I mean, it's worth remembering in and of itself, right, as just a general fact. But it's really important because when we do this cross and we're looking for funky stuff going on, be it linkage or epistasis, we're looking for changes in this ratio. So if we get a ratio that doesn't look like this, right, instead it's, what would be a classic one? One would be 12 to 3 to 1, for example, right? That tells us that something is going on which we're not accounting for. We're breaking one of our assumptions. And in that case, the assumption we'd be breaking would be the law of independent assortment, right? So one gene is affecting another gene, at least its expression, right? So that ratio is a useful kind of like marker, right, with which to compare the results that you get when you do this kind of cross yourself, right, if you're interested in that particular aspect. Okay, so anybody have any questions about dihybrid cross? We're going to break up for a little bit after this, just to restroom and coffee break and stuff. Does anybody have any questions? And again, hopefully the mixture of whiteboard and PowerPoint works. It is a little limiting working with such a tiny space. Well, if you do have any questions that come to you, just ask them after the break. Okay, so why don't we take a 10-minute break and get back here at 12:25, right? And, you know, kind of refresh yourselves, kind of stretch your legs, stuff like that. Stare blankly at a wall if that helps. Doesn't really matter. Just so you're good and fresh to go when you come back, because we're going to be doing some probability and statistics, right, which are non-trivial things to tackle. All right, I don't have a cat to take over right now. I've got my dog, but she'll probably follow me downstairs as soon as I get up. So I'm just going to leave this. Oh, here you go. We can stare at the whiteboard, absorb genetics facts. Okay, back in a bit. And remember, 12:25 we'll pick up again, okay? Hopefully that works now. What you doing up there, dog? Okay, so can you see the new PowerPoint? Just make sure. Awesome. Okay. This works a little differently between different video conferencing programs. Okay, so essentially there's two things that we need to go through here. One is the use of probability, right? So the reason why we're covering this now is that probability is a huge part of Mendelian genetics, right? Because again, the point to doing those monohybrid or dihybrid or test crosses is to use those assumptions to test whether or not what you think is going on is actually going on, right? And that's really where their strengths come from, right? Because you're not going to do a dihybrid cross, just for the sake of it. There's got to be some kind of scientific purpose for that. And so we have our assumptions from Mendel and from others about what we think is going on in those crosses, right? That's our assumptions. However, to be able to test whether those assumptions are correct, one, we need to work out what are the probabilities of getting those different outcomes in terms of offspring or phenotypes or whatever, right? Two, we need to be able to have a way of accounting for biological noise, right? So if you have, like the example that I gave earlier in the test cross of a heterozygous blue-eyed dog being crossed with a homozygous recessive brown-eyed dog, and that dog produced 8 pups. Now you might get 4 brown and 4 blue-eyed dogs, puppies, right? You might also get 3 and 5, maybe even 2 and 6, either way round, right? Because biology is noisy, it's random, and it gets noisier the smaller the numbers are, because essentially you just have individual random events having a much bigger, stop that, much bigger effect on the overall outcome. Those effects get smaller the bigger the numbers are, which is one reason why we like large sample sizes in biology. Anyway, so because biology is noisy and math is exact, right? Probabilities are probabilities. There's not like, yeah, maybe in there. It is a quarter, half, eighth, whatever. We have to find some way of figuring out whether what we observe, in biology terms, which is noisy, matches what we predict mathematically, right? And that's where our second part comes in, which is a goodness of fit test called a chi-square test, right? So both of those things are important. The probabilities are what give us our expected numbers or data. The chi-square test is what tells us if those our observed numbers differ in a meaningful way from our expected numbers, right? So those are really like the two big things that are going on here. And so when we talk about probability, essentially we got two different things going on. We have independent events and mutually exclusive events. So independent events are really ones where the outcome from one event has no effect on the outcome of another, right? So flipping a coin, rolling a die, whatever number or heads or tails you get in that one instance has no effect, or shouldn't, on the result that you get for the second, the third, the fourth, the fifth, or however many times you do it. Same with having kids. I don't know if any of you know people who have, had like 4 or 5 girls hoping to get a boy, right? Or vice versa, maybe, I don't know. They're independent events. What sex child number one is has absolutely nothing to do on what sex child number two is. They are independent random events, and so their probabilities are independent. And so if you, let's say you're hoping for a girl, and your probability of getting a girl, all things being random, right? Because that's our assumption, you've got a 50-50 probability or half or 0.5 probability of getting a girl. Now, are you as likely or more likely to get two girls? If it's a 50% chance of getting one girl, are you going to have the same probability of getting two? It's also a 50-50 answer, yes or no? No, yeah, right, exactly. Because essentially, the more events you look at, the probability of getting the same outcome in each successive event gets less, right? You can talk to, like my granddad was an avid bettor on horses for some reason. He used to look at the paper every Sunday. And it's kind of self-evident that your chance of winning on one horse is higher than winning on two horses, right? Because they're independent events. And so you have to take the probability of each independent event and multiply them, because that will give you a smaller overall probability for having both of those things happen. So if you want two girls, then the probability of getting two girls is going to be less than the probability of getting one girl, right? And if the probability of getting one girl is 50% or a half, then the probability of getting two is going to be a half times a half, which is a quarter, right? And three would be a half times a half times a half, which would be an eighth, and you can go on for as long as you wish, right? Does that make sense? Cool. So it's often said that phenotypes are independent events, right? So the phenotype you get is going to be, essentially, or the chance of getting a particular phenotype is going to be expressed as a fraction of all of them. So phenotypes are often referred to as being independent, right? And so the easiest way to think of a lot of these things is in terms of multiple events happening over time, right? So children, things like that. You can also put it into like a monohybrid cross, if you wish, right? So a classic question would be, if you have two individuals that have that are heterozygous for a recessive disease, right? Both of those individuals will not be expressing that disease. What's the probability that one of their children will express that phenotype and have that disease, right? So if you think back to a monohybrid cross, right? And both of those parents are heterozygous, so they're like the F1 generation. What's the probability of getting a homozygous recessive individual? Yeah. It's going to be a quarter or 25%. Right? So, okay, here's another question. What's the probability of one of their children being a carrier for that disease, i.e. being heterozygous? Now you think, yay, wow, hell, you're quick, Angela. It's going to be half or 50% because the genotype ratio is 1 to 2 to 1. Homozygous dominant, heterozygous, homozygous recessive. Right? So that's kind of where we're going in terms of thinking about this stuff in terms of probability, right? So the flip side of independence, right? So if an event is independent of another, the probability of both those independent events occurring is less than one of them. So it's going to be a multiplication. However, if they're mutually exclusive, you add them, right? So there's a good example. Here's a good example, right? So if you, well, let's just use this as this is a better example, I think, because this one's a little harder to explain. So let's say you want to have 2 boys and a girl, whatever reason. I don't know why. But let's say you do. Then you're going to have 3 independent events occurring. You're going to have your first child, your second child, your third child. However, you have more than one way of getting 2 boys and a girl, right? Hopefully that makes sense. You could have boy, boy, girl, you could have boy, girl, boy, or girl, boy, boy. Any of those orders will give you the exact same outcome. So for one of them, let's say the first one is boy, boy, girl. You're going to have 3 independent events, each with a probability of a half. Probability of getting a boy for the first, probability of getting a boy for the second, probability of getting a girl for the third. So because those are independent events, you multiply them together. So half times a half times a half is an eighth, or 0.125. But that's only one way in which you can get 2 boys and a girl. So you have 2 other options, each with the same probability. Now this is where we get to the mutually exclusive part. If you and your partner have 2 boys and a girl, and it's a boy, boy, girl, you cannot have had those children as a boy, girl, boy, right? They are exclusive event. You know, they're mutually exclusive. If you have a boy for the first child, that first child cannot be a girl. I mean, it sounds kind of obvious, right? I mean, that's, yeah. But it's really important in terms of probability, because when you have mutually exclusive events, the probability of getting one of them is an addition, right? So you have 3 different ways of getting 2 boys and a girl, 3 different potential orders. Those 3 different orders are mutually exclusive, right? You can't have one and then another one, right? You can only have one or the other, or the, oh, sorry, forgot about that, or the other one, right? You can't mix and match, right? It's either one or the other of the 3 of them. So the probability of getting any one of those is going to be an eighth plus an eighth plus an eighth, right? So you're going to have 3/8 or 0.375 probability of getting 2 boys and a girl, because you take those 3 series of independent events, and each one of those series is exclusive, right? You can't have this one and this one. It's either this one or this one or this one. And so the probability of getting any one of those is an addition, right? It's like if you think about, you know, firing a BB gun at an aluminium can, right? If you have 3 different cans lined up on the wall and you're not an especially good shot, you've got 3 times the probability of hitting a can, whatever can that might be, than if you only had 1 can on the wall, right? So that's why it's an additive probability, right? Does that make sense? Yeah, well, I hope so. I get better at explaining this every time I do it. Okay, and another way of thinking about it too is all of the possible potential ways to have 3 children should add up to 1. Oh, cool. I'm glad it does. I can't take any credit for that. That was my colleague. So if you think about, I'm not going to work through this, but you can if you wish, just to double check, right? What are the other options for having 3 children, right? You could have 3 boys. Probability of that is an eighth. You could have 3 girls. Probability of that is also an eighth. Yeah, there you go. Actually, you're missing some out there. You can have, partly because we have, yeah, and you can have 2 girls and a boy. But just as with the 2 boys and a girl, you've got 3 different options, right? So essentially, you have 3 possibilities for 2 girls and a boy, 3 possibilities for 2 boys and a girl, and then 1 for 3 boys and 1 for 3 girls. Each probability is an eighth. How many fingers am I, well, fingers and thumbs, digits, let's say, am I holding up? 8, right? So all of those will add up to 1, right? If you have 8 eighths, adds up to 1. So that's another way of kind of double checking your math to make sure that you're really thinking about this all the way through. So for example, Angela, if you tried figuring that out, you'd be like, hang on, I'm missing an eighth somewhere. Where's that other one gone? And you'd be like, oh, right, that's where it is. Right, so that's a good way of kind of double checking your logic, essentially. Okay, so now we're going to get on to the big one. Da, da, da. And let's see. I'm going to try and find. Oh, I could traumatize you with my excessive numbers of tabs. I've actually closed a few since yesterday. Let's make sure I pick up a good one, because last time I did this, I printed out a really crappy one, which didn't help anybody. Let's see if I can get this up nice and big. Okay, awesome. Yeah, that'll do. Okay, so let's go back to the PowerPoint. Just going to get that up, ready and waiting. So, as Mendel, if he was still around, would have been first to admit, his data did not match exactly the probabilities that he would have expected. Right, and so you can see some examples here. Let's move this out of the way a little bit. Here we go. Right, so all of these are the outcomes. These are the F2 generations, right, on the right two most columns of a series of monohybrid crosses, right? So we've got the P generation on the left, we've got the F1, and then we've got the F2 on and the third and the right and the fourth columns on the right. Now, as you can see, and we'll come back to these numbers, and you can use these to do some tests of your own as well, which I would recommend, right? You don't want to be doing this for the second time in an exam because you'll struggle, right? What you can see is that all of these should be a 3-to-1 ratio, right? According to the probabilities, according to the basic math. Oh, sorry. But what you actually see is that they're kind of a little bit, either side, right? And that's just based on random chance, like stochastic noise, essentially. And biology is noisy. It gives mathematicians absolute fits, to be honest. But there's just inherent noise in biology, just through random chance. And typically that noise gets smaller the bigger the population you look at. But still, even if you look at the biggest population possible for whatever thing you're interested in, you're still going to have some deviation from the mathematical probability that we calculate, right? And so really we've got to figure out a way of comparing these data, right, particularly the number of progeny, with what we would expect to get. And what we really care about is, is that difference due to random chance, biological noise essentially, or is it because something is going on? And that something will typically be an assumption which you shouldn't make, or which you made and is wrong, right? So again, going back to the whole Mendelian genetics things and those assumptions are key because they really underpin our analysis of those data. We're assuming we're going to get a 3-to-1 ratio because we did them on a hybrid cross, and that's what you should get, right? What we want to find out is that assumption correct, right? Even though the numbers, as 2.96 to 1, differ a little bit. Is that difference meaningful? Does it mean that something is weird? Or does it just mean that there's just some random noise, right? And so that's really an important point to get, because that's what we'll be talking about when we talk about p-values and our null hypothesis versus our alternate hypothesis. That's what essentially we're talking about, is this the chance of getting the same result again just by random chance, random probability. Okay, so hopefully some of you have done this recently. Come on, why are you going on to the next one? Come on. But we'll go over it again, assuming that you haven't. Okay, so what we're looking in terms of our statistical test is what's called a goodness of fit. And that really comes back to what I've said. How well do our observed data, right, these numbers, fit our expected data? And I'm using those words very deliberately because those are the same words we use in the actual chi-squared test. Right, so essentially what you're going to do is for every different condition, phenotype, whatever, you're going to calculate how different your observed data is from your expected data. And then what you're going to do is you're going to add those all together to make a larger number and divide that by what's called the degrees of freedom, right? Which is essentially how many different conditions do you have minus 1, right? And then that value and the degrees of freedom, you're going to stick it into a chart, right? And that chart, I don't actually know how they calculate it because they're smarter than I am when it comes to math. But we're going to use that chart to give us a p-value, right? So let's try and jump ahead a little bit here. So p-value is basically, you can think of it in two different kind of mirror images ways, right? It's the probability of that data being more extreme than what you predict with the null hypothesis, right? The other way of thinking about it, so that's basically saying what's the probability that you get some crazy ass stuff going on, right? The other way of thinking about it, which I kind of prefer, is what's the probability of that result occurring by random chance, right? For me, that kind of makes a little bit more conceptual sense, right? So, sorry, my dog's having a nightmare on lying on my bed. She's kind of moving her paws like this and grunting and whining a little bit. This is kind of distraction I don't get when I'm in the classroom, right? Anyway, so, and that matters because you are going to get differences from what you expect. But we want to know, we want to have some kind of number which says Yeah, this difference, it's just random chance. You do the same experiment again, and most likely you'll get a different result that's also not significant just by random chance. Now we have a lower cutoff, a threshold, which is really a somewhat arbitrary line in the sand, to be honest, right? And that threshold underneath that, and that threshold is 1 in 20 or 5% or 0.05, and I'll be referring to it in kind of decimal, so 0.05. Underneath that, it's more a case, that's saying you is that this result, this difference that you observe, did not occur by random chance. And if it didn't occur by random chance, something is going on, right, that you're not accounting for, that's breaking your assumptions or is interesting, right? Really, that's what we care about as scientists. What's going on and is it interesting, right? So if you get a p-value less than 0.05, what that's telling you is that there's something going on and this result didn't occur just by random chance. So if you repeat it again, most likely you would get a similar result, which will also still be different from what you expect, right? So that's kind of another way of thinking about it. Now, if your probability of getting it by random chance is high, that's basically saying that your null hypothesis, which means nothing is going on, still applies. It still holds, right? However, if you have a very small probability of that difference occurring by chance, then, sorry, Angela, I'll get to you in a second, then that basically means your assumption is wrong and your null hypothesis doesn't apply, right? So in our case, we're working with Mendelian inheritance, and so our null hypothesis is Mendelian inheritance is happening. So we should get a very similar number of when we do our observations to what we expect. If Mendelian inheritance isn't happening, then goes out the window, and we should get a much smaller p-value. Let's just see what Angela. Oh, okay, so just quick clarification for Angela. Above 0.05, that's essentially the probability that the result you got, the difference in this case between observed and expected, is due to random chance. So if you repeated this experiment again, maybe you'd get no difference. Maybe you'd get a difference, but in the other direction. It's just kind of noise, static, essentially, in biological terms. If you're below 0.05, that's telling you that the difference you see did not occur by random chance. So you have, let's say, a 5% chance that the result you saw was due to random fluctuation, just noise. That's a pretty low probability that it's random. So that's telling you, kind of as a consequence, that it's not random. Something's going on. Did that help? Okay. So another way of thinking about it as well is in terms of chi-square, and we'll get to actual chi-square in a second. Right, so in a chi-square test, what you're doing is you're adding up the differences between what you observe and what you expect. There's other jazz in there as well, but that's the general principle, right? So it's the sum of all of the real differences, right, in what you observe from what you expect for every different kind of condition. Pardon me. Now, if your expectations were correct, your hypothesis, your null hypothesis is supported, would you expect a really small difference between what you observe and what you expect, or a really large difference if your expectations are correct? You'd expect a small difference, right? I mean, you are going to get a difference most likely, and it will probably vary from one condition to another, but it will be a small difference, So let's actually, no, I'm not going to use this. Let's share my, I'll go away. My terrifying amount of tabs. Okay, so let's say we have two conditions, right? We'll get into the details in a second. That's degrees of freedom of 1, n minus 1. So if we had a very, very small difference, like less than 1, then most likely that difference is just due to random chance. You can see the bigger the difference, so we're going from left to right here from 3.8 under here. This is our p-value at the top, 0.05, 0.01, 0.001. And this in the chart is our chi-squared value, right? So as the chi-squared value goes up, like so, basically because the difference between what we expect and what we observe is getting bigger, then the chance of that difference being by just random gets smaller, right? So essentially what we're saying is that the bigger the difference, the less likely it is to happen by chance. How about that? Did that help? I don't know where my chat's gone. Let's get back to this. There we go. Oh, maybe. Kind of punching around a little bit. Oh, chat disappears again. Did that help? Cool. So yeah, so basically it's like if you're working as a maybe you're managing a bar, right? Let's say, and on the till, the cash register, as you're doing your counting up at night, it says you've sold like 100 beers at $4 each, and you get, out of the till, you get a net of $398, right? And so most likely, yeah, a couple of bucks might have fallen on the floor or something like that. It's just, shit happens kind of stuff. Now, if you do that and you get $300 back, instead of the 400 that you expect, that's a large difference. And so the probability that that large difference was just by random chance or accident is pretty small. Most likely, either one of your customers fiddled you or one of your bartenders did, right? Because the bigger the number, the bigger the difference between what you expect, what you observe and what you expect, the harder it is to explain that away just by accident or random chance. Right. And so if it's not accident or random chance, then the opposite is that something's going on. Right. And that's really what we're doing when we're using a chi-squared test in terms of goodness of fit. I haven't used that analogy before. Hopefully that helps. Okay, so there's going to be a formula here. I do apologize. Come on. But this is what a chi-squared test looks like. I'm never sure if it's chi-squared or chi-square. Probably seems to write it differently in different places. Doesn't really matter. Okay, so we got back, I mean, if you remember from working out variance for the lab, we've got this Greek letter epsilon here, right? This is, it means sum of. Oh, there you go. It actually says that in the slide. Right. So essentially for every single condition, if you have two conditions, let's look at this one, right? You've got round versus wrinkled. You've got two different conditions that you're looking at. You're going to work out this part of the equation. Hang on. Give me just a second. Come on. I just got to check something. I might have told you something wrong. Ah, come on, go away. Sorry, I can't get into my web browser for some reason when... come on. It's just playing silly buggers. Oh, yeah. Okay, let's go with that. So I gave you some... I don't know, I have to double check the audio, make sure I didn't mess that up. I'm getting a little confused about what I told you in the audio. I'll go back and check. But anyway, let's go back, get this up. Essentially, it's just the sum of those calculations, right? So if you've got two different conditions, then you calculate that difference for each one, right? So you would do the difference for round between the observed and the expected, and the difference from wrinkled as well. Now, a couple of things here. The very first one is you have to calculate the expected values, right? Can't do the thing without it, right? And so to calculate the expected values, this goes back to our probability, right? To our expectations if everything works out, our mathematical stuff. And so in a monohybrid cross for the F2, we expect that 3/4 of the offspring will be round, right? And the way we get because you can't work with ratios in chi-squared, you actually have to work with real numbers, right? It doesn't work otherwise. And so what you first have to do is you have to add those two numbers together, the number of round and the number of wrinkled. That gets you your total number of observed whatevers, seeds in this case. Now, to work out the expected, you're going to take that total, whatever that is. I did work it out last night, actually. Let's just do it now quite quickly. Oh, I can share that. That's funky. How cool is that? I'm learning stuff every day. What is it? 1850. Okay, so that's the total number of sample points, or seeds in this case, that you observed. You have to work that out because we're going to use that total number to calculate what you expect, right? So if your assumptions are 100% correct, right, and there's no biological noise, no randomness going on, you get exactly what you expect, then you should see 3/4 of this will be round, right? So you take this value, the total number, and now you take the probability of seeing one of these things, right? And that's how many round seeds you would expect to see, right? I think my voice come back from the shops. So 5493, I'll try and keep this in my head. To work out the first one, right? What we're going to do is, and again, this is harder to do on, yeah, that's not going to work online than it is in person. Right, so now we take the actual number, the observed number, which is 5474, and we work it out. So it's going to be 5474 minus 5493 equals minus 19. Now again, we can't sum negative numbers. This is the same as it was for variance. Oh, yeah, thank you, Andrew. So we've got to square that. And that gets rid of any negative symbols. And now we're going to divide that by the expected, right, which is 5493. Right, so that gives us this value, right? So this is our difference, our goodness of fit for the first condition. Yeah, that's right. Okay, does everybody get where I'm going so far? Right, so when we're looking at round, that's how much this number of observed round seeds differs from the number that we would expect. Okay, but that's only one condition out of two, so we need to do the other one as well. So I'm going to use that number that Angela put up, which is very handy. So we've got 7324. Hopefully you can see what I'm doing on the calculator. So we've got to work out our expected for the wrinkled seeds. Cool, that's good. Okay, so that's our expected number for wrinkled seeds. Again, if everything is perfectly exactly correct as we would expect, that's how many we should count. And obviously we're not going to, so how many did we count? Right? And so it's going to be 1850. Again, going back to the PowerPoint, that's the number of observed wrinkled seeds. We're going to subtract our, oh crap, I forgot what hang on, let's do this again. Memory of a fish times a quarter. Oh, there you go. I'm going to write it up here so I don't forget again. There you go. Hang on. 1831 expected. That's probably more useful. Okay, so we're going to do our observed, which is 1850. If I went back on here, yeah, too many things going on. I can't think of all this stuff at once. Okay, 1850 minus our expected, 1831 equals 19 again, which is kind of what you expect if you only have two different conditions going on. It gets more complicated if you have more, obviously. Square that, and now this one is going to be divided by the expected for wrinkled, which is 1831. Okay, so that gets us 0.197, right? So essentially, it's going to be 0.197 plus, because this is a sum of all of these calculations. And I think it's 0.06, give or take. Doesn't really matter too much. Right, and that's going to get us, oh, actually, let's do it for real. Get on over here. Okay, 0.197 plus 0.06. It's going to get us 0.257. So our chi-square value equals 9.257. Okay. Anybody got any questions about that? Like how I got that number? What the hell am I doing? That kind of shit. You're lost? That's totally fine. Where did I lose you? You can send it to me privately if
I
you want.
think,
I don't mind.
okay. Well,
No.
if we backtrack, you did the observed for the 7324 times the 0.25,
Okay.
and that gave you the 1831. But I'm
Correct.
not, where did you get the 0.25 from?
Oh, good question. That's an excellent question. So, in a monohybrid cross, what's the ratio of F2 progeny? That's a question to you, Gabrielle. Sorry for putting you on the spot.
Oh, okay. So was that 1/4?
Yeah. So it's going to be a 3-to-1 ratio, right, of the dominant to the recessive phenotypes. So you'd expect 3 out of 4 progeny to have the dominant phenotype, or 1 out of 4 to have the recessive phenotype. So that's why to get the expected value for the round, it's 7324 times 0.75.
Right. Okay.
Yeah, kind of.
Oh yeah.
Once you see it, right? It's kind of one of those. Until then, it's like, what the? And now we do the same thing again, right? For the other one, it's going to be 1/4, right? So the probability of getting wrinkled seeds is 1/4. So in theory, right, and this is theory because it's expected, we should get 1/4 of that number as wrinkled seeds. So that's why we times that by 0.25.
I got it. And then
Awesome.
so we when do we square it? Because you squared -19 as well up before that.
Correct.
Only when
So
it's negative number?
no, it's always. Let me write it out.
Okay.
Might be a little easier.
Yeah, I think calculator is what's confusing me.
It's all right. It's I'm going to stop sharing all of those so my picture gets a bit bigger. There. And I'm happy to spend as much time as is necessary going through this because this is a really both useful and important test that you'll need in biology. Okay, so apologize for my handwriting. Those are the two different conditions we have. Yeah. And so we're expecting 3/4 and 1/4, right? I know you already got that because you just told me so, but I'm just putting it up here for kind of clarity's sake and also in case anybody else has questions. Okay, so shit, I should write smaller. Our observed is, can't believe I can actually remember these numbers off by heart. Isn't that funny? Right, is that our expected, and I'm writing these up because this is really all the information you need before doing one of these things, right? So these are how we calculate our expected, right? Those are our assumptions, right? Those are what we're making our predictions with. Hang on. Yeah, it should be. Didn't you just put 7324 earlier, Angela? I'm trusting you here, man. It's important. Yeah, you did already. It's easy to get lost, right? Which is why it's so much easier just to write it out. Okay, so
Yeah, I'm so sorry. My question was the observed wasn't the total of the round and the wrinkled added together?
it is, which hopefully,
Oh, okay, I see. Okay.
yep. So
I'm just trying to match up what you're saying to the math that we're writing down, I guess.
yeah, so the total is 7324. So I kind of run out of space a little bit there. Hopefully you can see that on the right hand side. My webcam's also in a really weird place too. Okay, so observed for round, observed for wrinkled, total number of observed is 7324. Now our expected, we basically, here we go. We use that number and that number together, right? So 1/4 of 7324 is 1831, right? That's our expected value. And then we do the same for the other condition too, right? And that is 5393. Okay, does that all make sense? I'm not losing anybody. And that's not being mad or anything. I just want to make sure I got everybody on the same place. Okay, so what you want to do is you want to figure out.
And we were multiplying those, right?
Yeah,
Not subtracting. Okay.
just give me a second. So chi-squared, and actually let's keep using these colors because I think that might help. What you're going to do is the sum, right? I'm just going to put that there because it's easier than writing it. Is this one calculated? So it's 53474 minus the expected. Right? Squared. So you subtract your expected from your observed and then square that number. And the squaring is to get rid of any potential negative signs. At least that's how I understand it. Now, because you do it to one, you have to do it to the other one too. You can't just do it for the ones that are only for the ones that are negative. Otherwise, you'll get kind of weird, funky stuff going on. Does that make sense?
Yes.
Okay. Now, you'd have to ask someone else why you divide it by the expected, but you do. Ooh. So that's the complete calculation for your round seeds, right? So that's essentially going to calculate how much your round seed observation differs from your expected round seed number. Now, because there are two things going on, you've got like two halves, even though they're not 50-50, but you've got two different things that you're looking at. You have to repeat this calculation for this condition too, right? So let's do that one. So, that's a little bit more comfortable. That's going to be the same, right? Observed, right, which is 1850 from up here, minus expected, which is this one, squared. Just the same as you did for the other one, right? You can't do them any differently. I'm going to love those whiteboards when I get back to teaching face-to-face. And then you divide that by the expected again, right? So that's going to be 1831. Okay? So that, hey! But you have to add those together, right? Because you can't deal with just one alone because it's only part of the picture. You have to add both of those values together. So this is 0.06, give or take. It doesn't really matter too much. Plus this is 0.197, right? Unfortunately, I've run out of space because I don't have much space. And so that's the chi-squared value, 0.257, I think that says. I'm trying to read it backwards. Okay, so this is basically the sum of all of the differences between what you see and what you think you should see, right? But this is only part of the picture. It's not the whole picture. And that's because if you think about this, right, I'm going to put my pen back on my lid back on that. let's say you have 20 different conditions, right? So if you have the same kind of difference from what you observe and what you expect, you're going to be adding 20 of these things up, right? And so your chi-squared value is going to be bigger. So you need to take in account how many different conditions you have. Because the more the conditions, the bigger the chi-squared value is going to be, even if nothing is going on, right? Even if there's only a small difference, just because you're adding a whole bunch of them together. Yeah. So that's why we have to use a table, and we have to calculate, let's do it up here, the degrees of freedom. Which is always written as df. And that is, again, as we've seen before, n minus 1. So if you have two conditions, 1, 2, that's what n stands for, is how many different things are you calculating. In this case, we're calculating 2. If you're doing a dihybrid cross, you'd be calculating 4, right? So that would be the n in that case. And in each case, you subtract 1 from that number. So this is going to be degrees of freedom of 1, because it's going to be 2 minus 1. If it was a dihybrid cross, it would be 3, because it would be 4 minus 1, right? So you need this value and this value up here. And we're going to use both of those to figure out whether or not that difference is meaningful or not, right? Okay, anybody need to go back over any of that stuff? And I'm, again, happy to if you need to. If it's you're kind of not quite sure what question you want to ask, that's also fine. Think about it and then send me an email. Yeah, probably you'd have to send me an email or maybe even bring it up in lab later. It doesn't really matter, as long as I can answer it for you. Okay. I can hear someone tapping. Oh, n is, let's write it down. N is the number of different conditions. So a condition, got to spell condition right. So essentially, whatever you're measuring, each one of those things is a condition. So if you're, you have a trait where you have 4 different things that you're measuring, then let's say you're doing eye color, right? So you got brown, blue, green, and hazel, right? Each one of those is a condition, right? I'm sure there's probably a more technical word for it than that. But essentially, however many of these, no, not the sample size. That's a very specific term that doesn't really apply here. It's the same letter being used, unfortunately, which is kind of, a bit of a pain. But however many of these calculations you do, you take 1 from that number, right? So we've got 2 different calculations here because we've got 2 different things that we're looking at, round and wrinkled. So it's that number that you take 1 away from to get your degrees of freedom. Your sample size is that number. It could. I'm going to be a little cautious saying yes to that in terms of number of alleles, because it depends on what you're counting, right? So as a rough rule of thumb for this kind of stuff, yeah, I mean, that kind of works, right? So a monohybrid cross, you're going to get you have two different alleles, you have two different kind of phenotypes, essentially. Dihybrid cross, you got four different alleles, you have four different combinations of phenotypes. So that kind of works, but it can break down quite quickly. So a better way of thinking about it is number of phenotypes, right? So in this case, we've got two phenotypes. We've got the round and the wrinkled. In a dihybrid cross, you're going to have four different sets of phenotypes. You're going to have four different things you're counting. But we'll also, oh, so go on. Someone was going to ask a question. Perhaps they were just sneezing. That's okay. But you could also have situations where the number of alleles doesn't really track with the number of things that you're counting. So I'd be really cautious about using that as an explainer. Phenotypes is a much better way of thinking about it. But we're also going to be doing this in lab next Thursday with some actual data as well that you're going to collect from some pictures. Right, so you'll get some more practice. And we'll also be doing this again later on in lab with linkage too. So hopefully that should help. Now, let's just bring up. Okay, so remember we've got two different numbers, two things that matter. We've got the chi-squared value, which in this case is 0.257 on me. And we have the degrees of freedom, which because we have two different phenotypes, our degrees of freedom is 1. Come on. So this is one way of figuring out whether or not that difference is significant. And I'll show you a different way in a second. Let's just do this one first. So essentially you have three different things on here. We have your chi-squared value down at the bottom. That's one of your numbers. We have degrees of freedom, which are these purple lines going across here, right? That's the second piece of information that you have. And you're going to use those to figure out what your third piece of information is, which is your p-value over on the left-hand side of the chart. Okay, so to use this, you plug in your chi-square value, right, which is, let's just put that up again. Right, and that's kind of down here. I'm actually looking down there because it's like way down there on the far right. So let's just call it 0.2 just for simplicity's sake. Right, so now you follow that line 0.2 all the way up until it hits the line for your degrees of freedom. Right, I mean that's kind of, and essentially what you want to do is figure out what value is at that intersect, right? So you go all the way up here and now you follow the intersect across to the, let's do it more slowly, over to the y-axis, which is your probability, and that is 0.7, right? So your p-value is 0.7. So essentially what that's saying is that you have a 70% probability of getting that result by chance, which is pretty high. It means next time you've got another 70% probability of getting a slightly different result, again, by chance. And let's just put this up. And then p-value 0.7. Okay, so this chart isn't necessarily very intuitive, right? Once you figure out your way around it, it's not too bad. But another way of doing it is, Okay, and this is going to pop up this. I don't want to know about that. Fuck off. Come on. Go away. All right. Don't want that. Don't want that. Here we go. So the other way of doing it is this. Actually, let's find a fuller chart. There you go. This one's actually a little bit more useful. Here we go. Can you all see this okay? Awesome.
Yeah.
So don't worry about the numbers up at the top and the red box on there. It's just a chart to use. Okay, so what you do here is you find your degrees of freedom on the left, right? And our degrees of freedom is 1. And what you do is you go along until you find, this is kind of a weirder way of doing it, but that's fine. Like 0.257, right? And so that's going to be somewhere over here, right? And this isn't a very fine grain because really no one cares about this stuff above 0.05 all that much. So there's a whole bunch of missing numbers in here. But now you can say, okay, my chi-squared value for 1 degree of freedom, remember in this row, is between 0.02 and 2.7. And therefore, the p-value is between 0.1 and 0.9. And that's pretty crude, right? But at the end of the day, it tells us what we need to know, which is our p-value is greater than 0.05. And if it's greater than 0.05, then that result that we got, that difference between the round and wrinkled p's and what we expect, is not significant. It doesn't matter. It's just random noise. Our chi-squared value would have to go above 3.84, right? So to be able to be considered significant. So essentially you go towards the right, the chi-squared value gets up. And this column here, under the 0.05, your chi-squared number for that degrees of freedom would have to be bigger than this number for that difference to be significant. So that's just another way, and I'm going to stop sharing this so I can kind of get back to you lot. So that's another way of looking at the same kind of stuff. Essentially, you're going to have two things, your chi-squared value and your degrees of freedom, right? Chi-squared you calculated from the data and from your expected. Degrees of freedom you work out by how many different phenotypes or conditions you're looking at. And then you use those two figures, numbers, piece of information, to figure out what the p-value is, either using one of those charts or the fancy graph thing, which you see here. Right, and that's basically chi-squared analysis, right? So if and when I give you a problem like this to work out in your exam, which most likely I will, right? Because it's important to know and it's a big deal for the rest of the course as well. I will give you almost certainly that chart. I actually give you both. It doesn't really matter, right? Or you can use one or other of those that you find online too, right? Sharing files and stuff in Blackboard exams is not always easy. But you would use one of these to calculate your p-value, right? And I'd probably ask you, is it significant or not, for example? Or maybe what is the p-value? Or whatever, right? I might ask you a whole bunch of different questions. But you should be able to use one of these charts, either this one or that table, to be able to calculate the p-value from the chi-square value you calculated and the degrees of freedom that you calculated too. Okay. Does anybody have any questions? Oh, you're probably all a little bit wiped out, to be honest. It's been a bit of a tough morning study-wise in terms of information. Okay, so what I'm going to do is I'm going to finish this up and I'm going to need some lunch because I'm starving. I don't know about you lot. Let's meet again at 2 and I'll quickly go over the lab. Lab today is not going to take a lot of time. It's got, important and useful stuff to know in there. So it's worth doing, but it's not going to be very long. Okay, so hopefully it shouldn't take too much of your time this afternoon. So yeah, let's meet again at 2. I'll get the lab WebEx up and running a little bit before that, ideally. And then, yeah, we'll talk about lab stuff. All right. So see you in 20 minutes, give or take. I shall. I'm starving. All right, let's go.